So today we are going to talk about photosynthesis, which is a very interesting topic. So let's start with what is photosynthesis, which is the process by which green plants produce their food using carbon dioxide and water in the presence of sunlight. As we have it in this diagram, we have the carbon dioxide, we have water. So the plants make use of this carbon dioxide and the water molecules in the presence of sunlight to produce their own food, mainly glucose. At the end of the process, oxygen is given out as a byproduct. So let's talk about the equation of photosynthesis. You may be asked in two ways, either to write the chemical equation of photosynthesis or to write the word equation of photosynthesis. If you are being asked to write the chemical equation of photosynthesis, in this case, you have to write 6CO2 plus 6H2O map onto, you write on top of it light below chlorophyll, map onto C6H12O6, that is glucose, plus 6O2, that is oxygen. So in this process, we are making use of 6 molecules of carbon dioxide, 6 molecules of water to give us just 1 molecule of glucose. At the end of the day, 6 molecules of oxygen is given out as a byproduct. In the other case, in which you will be asked to write the word equation, what you have to do, you just have to write the carbon dioxide, not 6 molecules of carbon dioxide. Just write carbon dioxide plus water map onto. On the map onto, you write light, below it you write chlorophyll, map onto glucose plus oxygen. Now let's talk about the stages of photosynthesis. We have two main stages of photosynthesis. We have the light reaction stage of photosynthesis and we have the dark reaction stage of photosynthesis. So we are going to start with the light reaction stage of photosynthesis, which occur mainly in the thylakoid of the chloroplast, and the dark reaction stage, which we are going to talk about it later, it occurs in the stroma of chloroplast. So let's start with the light reaction stage, which occur in the thylakoid of the chloroplast. This process will begin with the absorption of light energy by the chlorophyll molecule. When the chlorophyll molecule absorbs light, the electron in the chloroplast, they become excited. When this electron become excited, they may tend to move. And on that process, another thing also has to happen. The water molecule in the leaf will be splitted into oxygen and hydrogen ion. Now, the process in which water molecule is being split into oxygen and hydrogen ion is what we call photolysis. Photo means light, lysis means to break. Now, these electrons that have been excited as the chlorophyll molecule absorb light, they may move along a series of electron transfer chain, like the plastoquinone that we have here, the cytochrome complex that we have on top here, and the plastocyanine that we have here. as the electron is moving along this electron acceptor or electron carrier, which have to accept the electron and carry it along this electron transfer chain, the electron may lose energy, and that lost energy will be used to transfer hydrogen ion from the stroma of the chloroplast into the thylakoid of the chloroplast that we have here. So what will happen as the hydrogen ion move from the stroma of the chloroplast into the thylakoid of the chloroplast, the hydrogen ion concentration in the thylakoid of the chloroplast will increase, because when photolysis occur, oxygen molecules are being produced and hydrogen ion are being produced in this process. And we say as the electron is moving along the electron transfer chain, it also lost energy, and that energy will be used to transport the hydrogen ion in the stroma of the chloroplast into the thylakoid of the chloroplast. As these two things happen, the concentration of hydrogen ion in the thylakoid of the chloroplast will increase. And we have an enzyme at the far end here, which we call ATP synthase. This ATP synthase, as the hydrogen ion which have become highly concentrated in the thylakoid, they may move back into the stroma of the chloroplast. As they are moving into the stroma of the ATP synthase will cause phosphorylation of ADP to form what we call ATP, adenosine triphosphate. The ADP is called adenosine diphosphate. This adenosine diphosphate will be phosphorylated by an inorganic phosphate to form what we call ATP, adenosine triphosphate. Now back to our point where we were talking about electron moving along this electron transfer chain, on which we have the carrier molecule, which I name it as plastoquinone, cytochrome complex, and plastocyanine. As this electron is moving, they lost energy. As we said, that lost energy will be used to move hydrogen ion from the stroma of the chloroplast into the thylakoid of the chloroplast. As the electron moves, what happens? They reach into photosystem 1, PS1. In the PS1, the chlorophyll molecule here, they absorb light at 700 nanometer, slightly greater than the one in photosystem 2, which is 680 nanometer. Now, when this chlorophyll molecule in photosystem 1 absorb the light energy, this electron which has lost energy as it was moving through this electron transfer chain may gain another energy. Now it may move by another carrier molecule which we call ferrodoxin. Now during this process of moving, NADP, nicotinamide adenine diphosphate, will be reduced to nicotinamide adenine diphosphate hydrogen, NADPH. Now at the end of this stage, NADPH and ATP will be used as the raw material in the dark reaction stage. But the oxygen here that is being produced during the process of photolysis will be given out as the byproduct of photosynthesis. So let's start with the dark reaction stage of photosynthesis. In the dark reaction stage of photosynthesis, ribose 1,5-biphosphate. It is called ribose 1,5-biphosphate because at carbon number 1, we have a phosphate that is being attached to it, and carbon number 5, we have another phosphate that is being attached to it. That's why it is called ribose 1,5-biphosphate. This ribose 1,5-biphosphate is a 5 carbon compound. The ribose 1,5-biphosphate, it is the one that accepts the carbon dioxide CO2 with the help of an enzyme called rubisco. Ribose 1,5-biphosphate carboxylase oxygenase. And during this process, after the ribose 1,5-biphosphate has accepted the CO2, many intermediate molecules will be formed which are very unstable. The first stable compound that will be formed is called 3-phosphoglycerate. So you also need to take note of the number. You can see we have 3 here in front of the ribose 1,5-biphosphate. And this structure you see here, which are off-white or which are black in color, they are carbon atom. And when you count it, we have 5. That tells you that the ribose 1,5-biphosphate is having 5 carbon atom. And the 3 here is telling us the amount of this ribose 1,5-biphosphate. So if we have 3 here, that tells you in total here on this part, we are making use of 15 molecules of carbon atom, as 3 times 5 will give you 15. But mind you, it is going to accept CO2, carbon dioxide, and we have 3 here in front of 1 carbon atom, as I told you. So it will be 15 plus 3 will give us 18. So in this case, we are making use of 18 molecules of carbon atom. Now, these 18 molecules of carbon atom here, as I told you, will form many unstable intermediaries. But the first stable one that will be formed will be 3-phosphoglycerate. Now we have how many carbon atom here? We have 1, we have 2, we have 3. 3 times 6 will give us 18. So we are still maintaining the 18 molecules of carbon atom as explained earlier. For the 3-phosphoglycerate to be converted into 1,3-biphosphoglycerate, it needs to make use of 6 molecule of ATP, adenosine triphosphate. And at the end of the day, that adenosine triphosphate will dephosphorylate. That means it will lose 1 phosphate to give us 6 molecules of ADP, adenosine diphosphate. And for the 1,3-biphosphoglycerate to form glycerate aldehyde 3-phosphate, it makes use of 6 molecules of NADPH. Mind you, this ATP and this NADPH, they were produced in the light reaction stage of photosynthesis. So for 1,3-biphosphoglycerate to be converted into glycerate aldehyde 3-phosphate, it needs to make use of 6 molecules of NADPH. This NADPH will be oxidized to give us 6 molecules of NADP plus. Why I say oxidized? because in reduction we are adding hydrogen ion to a compound or molecule. But if you are removing hydrogen molecule from a compound, we call that one oxidation. If you are adding hydrogen, it is called reduction. So that's why I say the 6 molecules of NADPH will be oxidized to give us 6 molecule of NADP plus. This glycerylaldehyde 3-phosphate is a 3-carbon compound. Now pay attention on this point, be attentive. As we have it here, we have 6 in front of the glycerylaldehyde 3-phosphate. Now what will happen? These 6 molecules of glycerylaldehyde 3-phosphate, one of it will be removed from the cycle to form glucose and other organic compounds. As we have here now, one in front of the glycerylaldehyde 3-phosphate. So one will be removed from that 6 to form glucose and other organic compounds. Now what will happen when we remove one from the 6? 5 molecules of the glycerylaldehyde 3-phosphate will remain. Now these 5 molecules of glycerylaldehyde will undergo a series of reactions using 3 molecules of ATP, which can give us 3 molecules of ADP, to form what we call ribose 1,5-biphosphate, which we talk about it at the starting of this dark reaction stage, that it is the molecule that accepts CO2. So we have various factors affecting photosynthesis. We have light intensity, we have CO2 concentration, that is carbon dioxide concentration, and we have temperature. Let's start with the light intensity. As we increase the light intensity, what will happen? The rate of photosynthesis will increase. But at certain point, even when you add or even when the light intensity is being added, the rate of photosynthesis will remain constant. There will be no increase in the rate of photosynthesis. Likewise, here we have in carbon dioxide concentration. When you increase it at a start, the rate of photosynthesis will increase. At a certain point, even when you increase the amount of CO2 in the leaf, the rate of photosynthesis will not increase. That's why you can see this line here going in a straight form here. That tells you the amount of carbon dioxide is being added, but there is no increase in the rate of photosynthesis. As we have in temperature, it varies with the light intensity and CO2 concentration. As you can see here, when we increase the temperature at first, the rate of photosynthesis will increase rapidly until a certain point where it will start to drop again back to zero. In this case, why the rate of photosynthesis is dropping is because higher temperature denature enzymes. It will destroy their structure, and these enzymes are needed for the process of photosynthesis to occur. So since these enzymes have been denatured, their structure have changed, what will happen? The rate of photosynthesis will not occur any longer, and in that case the rate of photosynthesis or the process of photosynthesis will stop. That's why the line, you can see it here, it is dropping back to zero. We have other factors that affect photosynthesis, like the amount of water present in the leaf, as water is also a raw material for photosynthesis. We also have the amount of chlorophyll molecule in the leaf, because it is the chlorophyll molecule that absorb the sunlight, and also the health status of the leaf. If the leaf is being infected, that one will also affect photosynthesis. So now let's talk about adaptation of the leaf for photosynthesis. This one is a popular question to WAEC, so you have to listen. And mind you, when we talk about adaptation, you have to name the part and you give the function of that part. That means how that part is helping the leaf in the process of photosynthesis. What is the role played by that part in the process of photosynthesis? So let's start with the first one here, which we call the waxy cuticle. Now this waxy cuticle, it prevents water loss but allow light to pass through. Mind you, we need water for the process of photosynthesis to occur, as it is one of the raw materials that we use in the process of photosynthesis. And light is also needed because the chlorophyll molecule absorb light energy, so it has to allow light to pass through. If the waxy cuticle was opaque, light will not pass through, and in that case the chlorophyll molecule will not be able to absorb the light energy for this reaction to start. And we have the upper epidermis. The upper epidermis, as we have it on the slide, it is transparent to allow light to pass through. So if this upper epidermis that is on top, if it is opaque, light will not pass through. And it is also a protective layer which prevents the entry of pathogen. Mind you, if the leaf is diseased, the rate of photosynthesis will be slower. A healthy leaf can perform photosynthesis at a higher rate than a diseased leaf. And next we have is the palisade mesophyll. These cells are closely packed together and they have high amount of chlorophyll molecule in order to absorb the light energy. They don't want this light energy to come and pass through without them being absorbed or without making use of it. That's why they are closely packed together and they have a lot of chlorophyll molecule to absorb the light energy. And we have the spongy mesophyll just below the palisade mesophyll. As you can observe it here, this spongy mesophyll that we have here, they have air spaces in it to allow free movement of carbon dioxide within the leaf, so that the cells of the leaf will make use of it for the process of photosynthesis to occur. As we have on the slide, it has pores called the stomata to allow carbon dioxide into the leaf, and it is the one that allows the byproduct of photosynthesis, that is oxygen, to be taken out of the leaf. And we have the xylem. The xylem brings water to the leaf. So when the root absorb water, that water will be transported into the leaf through the xylem. And we have the phloem, the one that transport the manufactured food. So after the process of photosynthesis, glucose will be produced. That glucose will be transported from the leaf to other parts of the plant, either where they are being used or where they will be stored. Because we don't want this food to be accumulated in the leaf up to the extent that it can affect the process of photosynthesis. So immediately this food is being produced, they are either transported to where they are being used or where they are being stored. So now let's talk about the differences between photosynthesis and respiration. In respiration, it occurs in all living cells. That means it is occurring in all living organisms. But photosynthesis, it is occurring only in green plants, not even all plants, but green plants. And respiration, it takes place in mitochondria. But photosynthesis, it takes place in an organelle called chloroplast, while respiration, it takes place in an organelle called mitochondria. In the process of respiration, oxygen is used. In the process of photosynthesis, oxygen is given out. And in the process of respiration here again, carbon dioxide is released. That means it is being given out. But in the process of photosynthesis, carbon dioxide is used. In the process of respiration, glucose is breaking down to release energy. But in the process of photosynthesis, glucose is created. It's not being broken down. It is synthesized or produced. And lastly, the process of respiration occurs continuously. Whether you are awake, day or night, this process of respiration, as long as you are alive, it is occurring continuously. But the process of photosynthesis, even though it is occurring during the day and during the night, but it is occurring mainly in the presence of light. That means during the day. Why for respiration during the day, during the night, it is occurring continuously. But in the process of photosynthesis, it is occurring mainly in the presence of light. That means it is occurring mainly during the day. Now let's talk about similarities between the skin and the leaf. This is a past question, so you have to focus on it and you have to listen well because it might repeat. The skin, it has an outer protective layer which we call the epidermis. The layer of the skin is divided into two, in some textbook into three. In some textbook in which they say it is divided into two, it is divided into dermis and the epidermis. In some textbook in which they say it is divided into three, they have an additional layer to the two which they call hypodermis. Hypo means below, so the layer below the dermis, and epi means above, the layer above the dermis. But in here we have to focus on the differences actually, let me not be distracted. It has an outer protective layer called epididymis, and on the leaf it has an outer protective layer called epididymis as well. And the skin, it contains pores, and the name of this pore is called sweat pore. And the leaf, it also contains pores, and the name of these pores is what we call stomata pore. And the skin, it is involved in excretion. In the skin, we have sweat glands that produce sweat. And the leaf, it is also involved in excretion, in which they have to take out oxygen, the byproduct of photosynthesis, through the stomata. The skin, it has veins. The leaf also, it has veins. Mind you, we have to talk about venation in leaves, how the veins are being arranged in the leaf. The skin also responds to stimuli, while the leaf also responds to stimuli. But the stimuli that the skin responds to is touch. So when you are being touched, you respond to it. But in the leaf, they respond to stimuli such as light. The leaf, it respond positively towards light. That means they move towards light. But it respond negatively towards gravity. That means they move away from gravity. And the skin, it regulates temperature through sweating. Likewise, the leaf, they regulate temperature through transpiration.